Today in these anatomy, microanatomy classes, histology, uterine tube and uterus. So this is we are familiar with uterine tube have got like a four divisible parts. We call them as infundibulum, ampulla, isthmus and intermural or interuterine parts. So whenever you are seeing that as an organ, as a viscera, these four parts are important. Largest of all is ampulla, where the fertilization takes place. And whenever you are seeing this particular organ histologically, it's a tube, but unlike the tube which is present in the gut, it has only 3 layers. Wherever you see in this gut which has got 4 layers, in this particular tube or fallopian tube we have got 3 layers. One is the inner layer, we call them as mucosa. So we have the lumen inside to it, then you have the mucosal layer, then we have a muscle layer, and then we have this adventitia layer. And again the mucosa, which is innermost layer, which is present in here, doesn't have that much of like a complexity, like it doesn't have that lamina propria or this muscularis mucosae, etc. That complex structure do not exist in here. Submucosa is absent, which is present in this like a GI system. We have seen that. So this particular fallopian tube consisting of 3 layers. Most inside we have the lumen to cover. Encircling that we have this layer, we call them as mucosa, and then mucosa is composed of the cells which is resting on it, like in this resting on this particular layer, which is like the lamina propria on it, or substance which is resting on this is lining cells. Then we have the muscle layer which is present in it, and then we have the adventitia. So this mucosa composed of these particular cells which is lining this particular mucosal cells. We call them as ciliated simple columnar epithelium. That means this particular thing which is columnar or cuboidal kind of epithelium. Predominantly we see that they mix of things like either column cells or these cuboidal cells present in it. So it has a little bit like a changes. Sometimes it is a little bit taller, it appears to look at columnar, but it's columnar to cuboidal cells which is present, and we have a cilia to it. Now the feature which this particular mucosa has, this particular mucosal epithelium goes on like a folding, or goes on like a I would say like it goes on like a magnifications. So we call them as longitudinal folds. So if you see this particular longitudinal folds, it is present in it, and under to it we do not have a submucosa, then straightway it is resting on the muscle layer and adventitia. So this cell which is present in here, we call them as ciliated columnar to cuboidal epithelium, and it is present with this particular like a layers which is thrown into the longitudinal folds. Then we have the muscle layer. We have this smooth muscle layered into two zones, I would say, inner circular and outer longitudinal. Then we have the serosa or outside layers. So basically it has got like a very simple networks: mucosa, muscle layer and serosa. The mucosa is having multiple folds, and it is lined by the cells which is simple columnar to cuboidal epithelium, and it is ciliated. And in between there we have the secretory cell, we call them as pigmentary or columnar pig cells. The mucosal, after that we have the muscle layer. We do not have any submucosa. Likewise we had this in a gut layers. This muscle layer have got two zones, inner circular, outer longitudinal. Serosa, which is outside most layer, which is lying by this peritoneum folds or peritoneal layer, which is mesothelium covering things. Now this mucosa shows numerous longitudinal folds. This is a kind of thing which is hallmark to identify. We call this pattern as a Farn pattern. So this is what you are seeing here, the cavity or lumen of this fallopian tube. Now this thing which you are seeing here, like a Farn pattern, just like fingers projecting into it, we call them as the longitudinal mucosal folds. So this thin layer is mucosa, and this mucosa goes into folding like elevations like this, just like villi, it is projecting out. Now this projections of the mucosa gives an appearance like finger-like or Farn pattern appearance, just like Farn, that primitive plant, that pattern appearance. So this particular layer we call them as mucosal layer. Then it is sitting right on this muscle layer which is present under to it. Then we have this muscle layer with a circular outer longitudinal, and most outside is the adventitia or serosa layer. So 1, 2 and 3 layers are there. So whenever you are seeing this particular thing which is Farn pattern, finger-like longitudinal mucosal folds, it gives a hallmark picture for this particular fallopian tube in sections. You are seeing that. So this is the actual picture how it should be looking like. You should be looking for the lumen. From inside you have this mucosa, then you have the muscle layer which is present, thick muscle layer, and then we have outside covering, we call them as adventitia. So nothing present between this particular mucosa to this particular muscle layer, absent of like submucosa, where you used to see the glands, blood vessels, etc. in the GI system. This is absent out here. And this finger-like projections which is going on, you call them as longitudinal mucosal folds, which is projecting towards the lumen. Now, what is the functions of this? This particular thing which is filled up with this particular secretory peg cells and like a supporting cells, which is called as columnar to cuboidal ciliated epithelium cells. This particular mucosal folds holds this particular ovum or fertilized zygote and allows this particular thing to move through. Now, we have seen that the fertilization takes place on some day and implantation takes place at some day. There is a gap between like 4 to 6 days. Between that time, from ampulla, it travels to uterine cavity and settles down. So, during that period, it needs some nutrition. Where from these nutritions will be coming? The nutritions will be absorbed running through this particular mucosal folds. So, this particular mucosal folds of this fallopian tube secretes this particular fluid which is filled up with these high nutrients or glucose materials, which is secreted by the peg cells or this particular like a non-epithelial cells. This nutrition is passively picked up by this particular developing embryo or ovum or zygote, whatever it is running through it. So, this is the purpose of this particular lumen to have this. Second thing is that it arrests this particular quicker flow of this particular zygote from this fallopian tube to uterus. Reason is that once this fertilization is taken on, we have to prepare our body accordingly that fertilization has occurred, and we have to prepare the bed for it, like the implantation bed that needs to be prepared, which is requires some time or some hormonal changes. So, we get this particular time through this particular passage, through this particular lumens, which is brushed by this particular longitudinal folds. So, this flow or this particular speed of the projections or speed of movement of the zygotes will be slower. Slowly it will be moving up. Meanwhile, this particular uterus becomes ready for receiving the implantations. So, this particular mucosal serves in two purposes: providing nutritions, holding or arresting or holding or slowing down the movement of the zygote or ovum from this particular fallopian tube to the uterine cavity. Now, this particular thing histologically, whenever you are going to see, we have to identify these finger-like projections of the mucosal folds, longitudinal folds lined by this particular cells is single layer ciliated columnar to cuboidal epithelium, and then we have this interspaced by the peg cells, which is secretory cells. These are not visible because these are microstructure, we cannot see them, but we have to know theoretically. And it is mucosal cells is resting directly on this muscle cord, and then we have the adventitia, which is outermost cord. So, these are the hallmark to identify this. Schematically, it should be looking like this. In the real picture, this is again zoomed out like 10x magnification. It will be seeing that in whenever you are going to see this particular like a microscope in real pictures, it will be like a little less magnified than that of this particular pictures. So, this is about fallopian tubes. It's simple as that. So, you have to remember these particular charts related to the fallopian tube. 3 layers: mucosa, muscle coat, and the serosa. Mucosas containing of this particular epithelium cells, columnar which are ciliated, maybe some cuboidal cells are there as well, interspaced by non-ciliated cells, we call them as peg cells. The functions of the peg cells is to provide the secretions. Then muscle layer, which is having 2 layers, inner circular, outer longitudinal, and this particular serosa over there. The hallmark for this is that mucosa is going on longitudinal folds or finger-like folds, which is looking like fern pattern, finger-like projections, fern pattern, finger-like projections seen like this. So, this is how we are going to identify this fallopian tube. Next, moving to the uterus. So, the different parts of the uterus, the gross anatomy we know by this part, the parts which is present, body, fundus and cervix, and this canal which is present inside to it. It's have got like a layers which is present inside to outside is known as endometrium, myometrium and perimetrium. So, likewise, whenever you are going to see this particular cross section picture, you are going to see these particular layers like endometrial layer, myometrial layer, and outside perimetrium, which is the serosa layer. So, the endometrium is the innermost line which is layering over this cavity of the uterus. So, it is present inside the cavity of the uterus. We call them as endometrium, which is having some kind of cell lining, which is lined by this simple columnar to cuboidal epithelium. And this particular lining epithelium, which is not ciliated compared to the previous one, it's not ciliated. And then it has a plenty of glands which is present in this particular layer or innermost layer. So this particular layer or innermost layer, endometrium, contains two things. One is lining epithelium cells and second is plenty of glands are there. We call these glands as a uterine glands. Depending on stage of that particular uterus, we have this particular glands taking different shapes. Like we have this particular uterine cycle runs like 28 days. We have this particular shredding phase where you have menstrual bleeding going on. Then we have this particular thing goes on the delivering into proliferative phase, early proliferative phase and secondary or proliferative phase. Then it has a secretory phase and then it has a menstrual period which is going out. So depending on how you are picking up that particular uterine specimen, this particular gland or which is present inside give a morphological changes. We will be coming into it, but it contains this innermost layer lining epithelium with the glands which is present, and it is very visible and very hallmark for this particular organ, like endometrium layer which is having filled up of this glands which is very visible. It's a kind of like very distinct kind of histological presentations, and this gland also lined by the simple columnar to cuboidal kind of epithelium. Then we have the muscle layer which is present in here. We have a thick muscle which is present to it, inner longitudinal, middle layer which is irregular or crisscross arrangement is there, and outer longitudinal. But to cut short, it's a middle muscular layer, and then outside layer which is the covering layer with serosa layer. Now, this particular endometrium, depending on the stage of that particular uterine cycle, can have a depth or can have some kind of breadth. So if it is present in the menstrual period, this particular endometrial breadth is less. If it is in the secretory phase, it is more. Why? Because it goes for this particular histological changes into the endometrium. So we call this as a proliferative phase, where this particular after menstruation, this particular uterus or endometrium grows under influence of the hormones. Like everything inside the glow, cell grows in size, the gland which grows in size, and so is the blood vessel which is present in here grows out here. So we call this as a proliferative phase. Then we have the secretory phase where we have the apex reaching to the growth of here. So it is in this particular secretory phase, that particular growth of the endometrium is maximum. That means it reached to the apex with the highest level of hormonal stimulation. The glands are super big. The endocrine glands which is present, exocrine glands which are present in here are very big, and this particular blood vessels, they are very big and tortuous in nature in this particular regions, and as well as the cells are big in this particular phase. Then it reaches to this particular phase or shedding off. When this particular hormone cuts out, this particular cells and cellularities and this particular vascularities go down, and there is a necrosis. After that, it is shedding out of the cells and this particular thing drops down. So there is a constrictions of the blood vessels, then you have the narrowing, and we have these enzymes which is pulling or scavenging this particular regions of the endometrium. So this particular thing, we call this as this menstrual phase of this endometrium. Depending on which phase we are picking up this particular endometrium, we get this particular changes. So glands, for example, in this early phase or post-menstrual phase, we are getting a gland which is like this, which is a tiny one. This particular same gland, with the influence of the hormones, moves into bigger size in the proliferative phase or late proliferative phase. Once it reaches to the second phase, we call them a secretory phase, it becomes super coiled, super large. See that it becomes large, it becomes coiled and becomes voluminous, it secretes plenty of fluids from it. So this is the highest point where it is going, and then it all of a sudden it loses this particular hormone and it loses this particular growth. The blood vessels also shrinks. This particular gland destroys and reaches to this again this particular stage. So whatever the growth that this gland has made is shed off during the periodic shedding off of menstrual period. So this particular glands which you are going to see will be on different size and shape depending on how this particular uterus has picked up whenever this particular staining was done. So normally you see that this is the phase where you can see this particular growth is layer which is less. So this is the inner layer, this is the lumen side, this is the endometrial lining epithelium, you see that, and then we have these glands which is present in it. Then you have the muscle layer and then adventitia. This is how it is oriented. Now the entire length of these things is endometrium. The thickness of the endometrium is quite big. You can see that. Now this particular thing is covered with this particular glands which is present and blood vessels which are present as well. So this area which is endometrium or innermost layer is present of the lining epithelium cells which is cuboidal kind of epithelium, Then we have the gland, which is lined by the cuboidal kind of epithelium, which is present in this particular layer, and we have the blood vessels. This is an early phase of like a early proliferative phase. Then it moves to the next phase, which is proliferative, late proliferative or secretory phase. The same thing, we can see that this is, for example, 5 millimeter, and this has become nearly 8 to 9 millimeter. So endometrium, the breadth of the endometrium has grown bigger because of the influence of the hormones. What has grown? Everything has grown in this particular phase. This particular glands which you seeing here becomes enlarged, tortuous, voluminous. The blood vessels which was there, it's becoming large and becomes spiral, and it holds lot of fluids inside to it, and this particular thing it is getting lot of kind of space inside. So this is reaching to this like a late proliferative to the secretory phase from this early proliferative or post menstrual phase. So this is how this particular changes occur. Normally, whatever the slides we are picking is usually like in this particular late or like early proliferative phase, we usually get this particular specimen from. So we have these particular regions, which is endometrium, which is present over here. Under this endometrial cells, we have this lamina propria, which is occupied by this particular plenty of the uterine glands and blood vessels inside to it. Then we have the muscle layer, then adventitial layer, which is following here. So this particular picture, you are seeing the real picture, how this particular thing it should be looking like. These glands are visible in here, early proliferative phase, and then we have this late proliferative phase, and then which you can also call as a secretory phase. We can become tortuous, enlarged like this, and the muscle layer more or less goes same. So this particular thing, as far as this particular like phases of this particular endometrial shedding, we can call this particular uterus into few zones. Now we call this particular endometrium based on this how this particular hormone is influencing into 3 different zones. The lowest most zone we call them as stratum basale. This particular zone is lying on right top of this muscular zone. Then we have the middle layer, we call them as stratum spongiosum and stratum compactum. So these layers are going from the top of middle layer, and this is the superficial most layer. Whatever the changes which is happening in case of the proliferative phase or in the secretory phase, this particular regions we call them as stratum compactum and stratum spongiosum that manifests. So whatever the changes you are seeing here, these particular changes which you are seeing here is predominantly happening in this layer and in this layer. The less changes is presenting on this layer, which is known as stratum basale. So this basale is rather lying at the base of the endometrium. This is shows little changes. More we move up from this particular lumen side, from this particular base side, it shows more and more proliferative or secretory changes. So this layer we call them as stratum basale, and then on top of that we have the stratum spongiosum and compactum, which undergoes maximum number of the changes which happens because of the hormonal influences. So to summarize the thing, whenever you are going to see this histologically, we are going to see 3 layers: endometrium layer, myometrium layer and a perimetrium. Endometrium we are focusing out here most of the time, which is a large depth of endometrium we see. We have this lining epithelium, columnar to cuboidal cells, connective tissue which is present with the lamina propria occupied by the tubular glands, and many of the arteries are present, and myometrial muscle cells are there which is having different directions on it, and perimetrium most outside there. So whenever you are focusing this particular histological slide, we are going to see this endometrium in great detail than that of rest of the layers. And based on this particular endometrial changes, whatever we are seeing that most of these striatal changes is going on this particular stratum compactum and stratum spongiosum. Least changes is happening in the stratum basale. The hormones which are influencing is estrogen and progesterone, which you understand in the physiology class better. Initial phase we have the estrogen, and later on this progesterone takes over for these particular changes which is bringing on, and later we cut this particular progesterone and estrogens because of ovulation happening with this fertilizations or not. After this no fertilization, there is regressions of the progesterone and estrogen, and then we have this particular everything is shading off. This particular stratum compactum and spongiosum is shades off. We call them as the menstrual phase or menstrual period. So we get this 3 menstrual period stage like proliferative phase, initial phase or post menstrual phase. Then it goes in the highest level of this enlargement, we call them as secretory phase, and then we have this menstrual period which is falling on these regions. So this is how we are going to identify. So we have the two slides are there. One is for these, two of them for these fallopian tube and two of them are for uterus. Both of the slides are very easy to understand. Once you understand this things related to this, understanding these histological features about this uterus and fallopian tube.